Hello, NEET aspirants. So today we are going to start with the chapter Body Fluids and Circulation. Before starting this video, I would brief that I have highlighted the important points in the colors, as well as I have made a short notes out of it, so which will help you to understand the concepts better. So let's start with today's chapter, that is Body Fluids and Circulation. As you know that body needs nutrients, oxygen, essential substance. At the same time, it will utilize all this and it will have waste product and harmful substances which has to be expelled out of our body. So why this is important is for proper functioning of tissue. So different animals have different method, okay? For example, this sponges and this coelentrates, which are nothing but aquatic animals, will use the surrounding water, okay? Human beings use blood, as well as lymph also helps in the transport of these substances and to expel the waste product. So let's see here. So this is sponges, which will use the surrounding water, as we know, which has oxygen, which will utilize, and it will utilize, and after utilization it will expel carbon dioxide. In the same way, human beings have blood for the circulation of oxygen, as well as to take carbon dioxide from the body and to expel out. Next we are going to learn about the blood. So blood mainly contains plasma, fluid matrix and formed elements. So plasma constitute 55% of the blood. It mainly contains water in the ratio of 90 to 92%, and it contains nutrients like glucose, nitrogenous waste, lipids, gases like oxygen, carbon dioxide, electrolytes, protein. Protein constitute 6 to 8% of the plasma, and the proteins present in plasma is mainly albumin, fibrinogen, globulin. Looking at its property, albumin is mainly important for osmotic balance, fibrinogen for clotting, and globulin for defense mechanism. Okay, and it has fluid matrix and formed elements. Formed elements constitute 45% of the blood. So it is mainly divided into WBC, which is also called as leukocyte, platelets, which is also called as thrombocyte, and RBC, which is also called as erythrocyte. So WBC is further divided into granulocyte and agranulocyte. So examples of the granulocytes are neutrophil, basophil, eosinophil. Agranulocytes are lymphocyte, monocyte, and plasma also contains clotting factor, but in the inactive form. It is activated only when there is any injury where there will be blood coming out of the body. And note that plasma, if you eliminate or remove the clotting factor, it is called as serum. So let's see here how you will separate this constituent is through method called as, you can see here, centrifugation. So when you do that, you can have different components of blood separated. So you have plasma, which is nothing but a straw colored, and you have white blood cells and platelet here, and the red. Moving up next, we are learning about the formed elements that constitute of 45% of the blood. So formed elements are mainly RBC, WBC and platelets. First we learn about RBCs, which are also called as erythrocyte. In blood, if you measure, it will be 5 million to 5.5 million per mmQ. It is mainly formed in bone marrow, and it is destroyed in spleen, which is also called as graveyard of RBCs, and its lifespan is about 120 days. So if you take 100 ml of the blood, you will get 12 to 16 gram of hemoglobin. Now you might have a question, what is hemoglobin? So hemoglobin is an iron containing red color pigment which is present in the RBCs. Hence, since it is red in color, it gives an color to the RBCs red. So it is called as red blood cell. And RBCs don't have any nucleus, and it is biconcave shape. Next we learn about WBC, which are also called as leukocyte. It does not have nucleus, and also it does not have any hemoglobin. Hence it is colorless. In blood it is 6000 to 8000 per mmQ. Its lifespan is shorter. If you classify it, it is divided into granulocyte and agranulocyte. The granulocytes are neutrophil, basophil and eosinophil. Agranulocytes are lymphocyte and monocyte. In this, most abundant is neutrophil, least is basophil. Neutrophil is around 60 to 65% of WBC, whereas basophil is 0.5 to 1% of WBC. Now, looking at its function, neutrophil and monocyte both helps for the phagocytic property. That is, it will phagocyte or destroy the foreign organisms entering into the body. whereas basophil is involved in the inflammatory reaction. Eosinophil will prevent the infections as well as involved in the allergic reactions. So basophil is involved in inflammatory reaction by releasing histamine, serotonin and heparin. What is this inflammatory reaction? It is nothing but a body defense mechanism. For example, when you get injury or any infection, you will have redness, swelling. All those things are inflammatory reaction, and mainly because of release of histamine, serotonin and heparin. Next, moving ahead with lymphocyte. Lymphocytes are of two types, B and T, so which are involved in the immune response of our body. Then moving ahead with monocyte. Monocyte is around 6 to 8%. So you can see here in this diagram that RBCs are not having any nucleus. Platelets, while has eosinophil, you can see that the nucleus has two loops, whereas in the neutrophils you can have multi loops. Next we have platelets, which are also called as thrombocyte. How it is formed? In the bone marrow we have megakaryocytes, which are fragmented to form platelets. In the blood we have 1,50,000 to 3,50,000 per mm3. So why this is important? It releases substances very important for clotting or coagulation of the blood. What does that mean? Once if there is any injury or hurt, there will be bleeding from the injury site. After some times there won't be any blood oozing out from that site because there will be coagulation or clotting of the blood being happened. So if the number of platelet reduces, what will happen? Instead of clotting, there will be excess loss of blood. Next we move upon with blood group. So you have mainly two type of blood group. One is ABO, another one is RH. So before understanding the concept, you should know that there are two types. That is, one is the antigen, one more is the antibody. What is this antigen? You know, this is an RBC. On this surface you will have antigens, that is A and B. Okay, what this will do? Whenever it enters your body, it will induce an immune response, while proteins produced in response to this antigen is antibody. So for example, if there is a blood mismatch, so this antigen won't be matching the body, so this will trigger the immune response, and against that antibody would be formed. That is what been written here. Matching is important for transfusion. If not match, what will happen is that antibodies will destroy the RBCs in the body and there will be something called as clumping. Carefully observe this table. So you have different blood groups, that is A, B, AB and O. So this will have this many antigens, but you know that O doesn't have any antigen. So if you give this kind of blood, that is O blood group, to anyone, it will not trigger immune response in anybody because there is no antigen only. So blood group O can donate blood to any group. So hence it is called as universal donor. Now suppose you gave this blood, these different bloods to someone. Something has to be produced in return if it is not matched. But for this AB blood group, there is no antibodies produced. Okay, so it can accept blood from any group. Even if it accepts blood of any of this group, it has no capacity to produce antibody against it. So it can accept blood from the any person. So it is called as universal recipient. After this ABO blood grouping, you have other blood grouping like RH grouping. That is, RH antigen is present in the RBCs in 80% of individual. If it is present, it is called as RH positive. If it is not present, it is called as RH negative. This was first seen in the rhesus monkey, hence the name RH. Now moving ahead, we have erythroblastosis fetalis. What is this? So if a mother is RH negative, but baby is RH positive, how did this happen? Because father was also RH positive. So since father was RH positive, baby got that RH positive from its father, not mother. Mother is actually RH negative. Now what will happen? In first pregnancy, there will be no problem. Delivery will happen, but during delivery, through the birth passage, there might be mixing of blood. Now what will happen? This RH blood group of that baby, RH positive blood, will enter the mother. Okay, so it will enter the mother. Now the delivery has already happened, so no effect for the first pregnancy. So now the mother will start producing antibody. Now in the second pregnancy, mother has already produced antibody. Now this will enter the fetus and it will break the RBCs of the fetus. So what will happen in the fetus is there will be anemia, that is nothing but reduction in the RBCs that will affect the circulation. And what is jaundice? Jaundice is nothing but the breakdown product of the RBCs, that is nothing but increased bilirubin. So how to prevent it? That is after the first child, give anti-RH antibody. That is, see, you can't avoid the complete mixing of the blood into the mother's circulation. There might be some mixing. So if some mixing has been happened and if you give anti-RH antibody, it will kill that RBC and prevent the immune trigger of the mother. Next, we learn about coagulation of the blood. You know that after an injury, there will be bleeding from that site. But after some times, what happens is there will be a dark reddish brown scum would be formed, but there won't be any bleeding. This is because coagulation or clotting might have happened. So what is this scum? It is nothing but network of thread-like structure called as fibrin. Okay. So plus it will contain damaged formed elements trapped in it. Okay. Trapped in it. And how this is formed? This is by the activation of inactive forms present in the plasma. That is, prothrombin would be converted to thrombin first, that is through the help of the enzyme thrombokinase. Further, this thrombin will help in the conversion of fibrinogen to fibrin. This fibrin will form that scum and block the leakage site from that site of injury. Next, we are going to learn about lymph or tissue fluid. Before understanding that, you need to know that from the heart, oxygenated blood is carried through the artery into the body. In the body, arteries become smaller and form capillaries. Then deoxygenated blood, because from the capillaries, oxygen would have utilized by the body. Only deoxygenated blood would be left. It is carried by the veins into the heart. Now at the level of capillaries, as you can see, blood is entering in the capillaries. But what will happen is water and water soluble substances move out from the capillaries. Okay. So this is the space between the capillaries cells. So you have this space. So what is this? This is nothing but called as interstitial fluid or tissue fluid. This interstitial fluid, whatever is present, has the same minerals as that of plasma. Whatever the plasma in the blood, whatever it contains, that is nothing but the minerals, the same minerals is present in this interstitial fluid. It is an exchange media. Whatever the blood contains nutrients, oxygen, first it will be transferred to this space and from this space enters the cell. So it is a transport media present in between the capillaries and the cells. So what will happen, how this fluid will be eliminated or transferred? So from here, from the space, this space, it will enter now lymphatics. Once it enters lymphatics, it is called as lymph. Later it is carried into major veins and transported to heart again. So what is this lymph? Lymph is nothing but the fluid present in the lymphatics, that is from this potential space containing water and water soluble substances. This lymph is colorless and important for immune response. It also contains nutrients, hormone, and in the intestine you have villi, intestinal villi. It also contains lacteals, that is nothing but lymph vessels, from which fat is absorbed in the intestine. If you find my videos useful, please do like and subscribe. Thank you.